I think we should get started. So this is a panel entitled Digital Technologies for Clinical Care. And the objective of this panel is to discuss, what are the important questions when it comes to digital technologies in the context of clinical utilization, and what we can do also as a multi-campus enterprise to enable development of such technologies. So we have three panelists today. I think we're going to ask them to introduce themselves.
Yes. Good morning, everyone. My name is Alex Segares, and I'm a Senior Research Associate in Computational Biomedicine at Weill.
I'm JP Pollack. I'm a Senior Researcher in Residence at Cornell Tech, Assistant Professor of Clinical Epidemiology here at Weill, and co-founder of a group called the Commons Project.
Good morning. My name is Cora Sternberg. I'm a postdoc associate at ICB, and my research focuses on clinical image analysis using AI.
And so, please.
Hi, I'm Deborah Estrin. I'm a professor of computer science at Cornell Tech and started a health tech hub. And I'm here in the healthcare policy and research department, and a close colleague of Tanzim and JP, who you heard from yesterday.
And so Deborah and I have been charged with moderating this panel, coming up with questions. So I think we'll get started with questions for the panel. So one of the questions was, what do you work on that has significant impact on patient care, and in what context? When it comes to things like tracking and predicting patient behaviors, image analysis, computational drug design, EHR integration. How does the work that you do employing digital technologies impact patient care at this moment, and how do you see the evolution of this impact in the future?
I can start. So I build all the software tools that we use in our clinical genomics assays at Eipm. And this essentially involves from the moment that clinician orders a test all the way till they receive back the report. It involves the LIMS that we use on our daily basis on the lab to track and essentially process samples, to the business intelligence that we have in our pipelines to automatically fire, to the AI-assisted precision medicine knowledge base that our molecular pathologists use in order to sign out the cases. And the last segment, it's the plumbing that actually gets into the EHR.
So I work on a range of tools ranging from sort of infrastructure, things like Research Stack, which is a tool set for building mobile health research applications, to projects like Common Health, which is the Android equivalent to Apple Health to try and bring clinical records down to individuals' phones. And last, actually working with clinical collaborators to build specific apps to go into either research studies or to care. Most of this work is kind of centered around this premise that we and our patients all have mobile devices now with us 24/7. And the fact that we can leverage those devices, both for data collection and for delivery of intervention, is something that we absolutely must be taking advantage of, and there's no excuse not to.
So our team research focus on clinical image analysis. We started with pathology images, and we extended our project to other type of images like MRI embryo. You may heard about that. So specifically, we develop new frameworks based on the deep learning algorithms, such as convolutional neural networks, to classify the images for different condition, for different diseases. For example, in one of our research, we developed a framework that could classify two subtypes of non-small cell lung cancer, squamous cell lung cancer versus adenocarcinoma, just based on the images and the pathology images. And also in another projects, we try to classify the aggressive versus non-aggressive prostate cancer based on the MRI.
Maybe just one comment. It looks like the technologies that you guys have discussed, obviously are great, and on the research side, have achieved a lot of performance, if you want. There seems to be a gap in terms of clinical implementation and clinical utilization. How do we fill this gap? And Deborah, feel free to also chime in.
So to me, the most difficult part of that, the pre-printing label data. So it seems that when these images, for example, digital pathology images or MRI was captured and saved, they didn't think about the future projects or AI. So we have hard time to find the correct labels. And that's it. I think it's, to me, as the most difficult part is labeling and annotation that I really wanted, the correct one. So that's it. I think it's for us.
Go ahead.
I was just going to chime in answering your question. So I think it's a great one where there are gaps to clinical, and there's a certain amount of sort of just parallel play. These three things are going on and are not particularly integrated. So for us, it's also perhaps a data problem, but it's not that the data exist for the most part and aren't labeled. It's that for the most part, we just lack this. We're not capturing the data from the patients as they come in and go through clinical care, let alone using that data in clinical care. And it's just, the devices are just sitting there in their pockets. They're generating data to be used for advertisements and algorithms. The data are there, and the clinical care is there, and there's no connection between them because it's not so easy. We have to actually figure out how to validate how to use it effectively in clinical care. Just as with precision medicine of any kind, it's like you need to not have to have a fleet of PhDs behind the clinician as they're trying to advise. And we have that same problem. That's okay, except we've had that same problem for 10 or at least 5 years. And so, yeah, it's hard to get it started because it's catch-22.
Yeah, just picking up on that latter challenge that Deborah described. So one of the things that I see all the time is we as information computer scientists, the really interesting parts of a lot of the problems are, or at least in my line of work, is the really early stage work, right? It's getting the technologies, working with a clinician to develop them, to get early pilot data, to understand how they might work in the patient context. And the next phase, which is the actual validation of that, becomes a little bit less interesting and more out of our domain. So there needs to be sort of follow through on the clinical side, and there's not often funding mechanisms. There's no obvious place to go when you say, okay, I have a really cool pilot app, and now I need to get enough data before I'm ready to actually go get an R01, right? So where things fit in that and where things get lost is something I see all the time.
One thing to add, or at least my strategy on closing the gaps, is essentially finding the right people in the room and start connecting the dots. So with JP and Deborah, we've been collaborating together on a trial for patient-reported outcomes on mobile health for our cancer patients. And we've been essentially using their domain expertise and ours and combining it to have a better outcome with Pega and Iman and Likika on the back. We've been working together on embryology and trying to essentially combine AI and mixed reality together to visualize all these embryos and have the AI being the backbone on analysis. So there's always gaps in the field, and essentially precision medicine has a widespread and opportunities to work together to start connecting these dots.
So now I can ask a question, but interject something in my question. So listening to some of the talks yesterday, Isaacson's, for example, and the talk earlier this morning, the first one of the day, there's clear desire and need for the science to be driven forward, perhaps not the pathology, but for even the translation of that into clinical. There is need for more information from the patient. And if we could identify where those biggest gaps are, because yes, everyone says yes, but clearly the need isn't large enough to have actually made the effort to start to get it. And so if we can identify, as JP was saying, where we have high priorities that maybe intersect, maybe we can make more progress. So the question that I should be asking you is, what would you like to be doing? Like, there are the things you're doing, and you mentioned some. You wish there was more labeled data. You could be doing other things. But if there was a project that you could be doing now that you see as feasible and truly rewarding, what would that be?
I can start. I'm obviously biased to pick AI and spatial computing, which is the part of mixed reality. And for me, it is how we can empower each clinician or each scientist to be able to tap on the big data that we're producing, that Deborah is talking about, and visualize it in a completely novel way to convert that big data into knowledge with AI, and from knowledge convert it over to actionable items. And this is not just a specific disease group that I would like to work on. This is multifaceted. So I talked about embryology. We were looking yesterday at Dr. Zbana's presentation on how we use spatial computing on a lab setting. There's so many different work groups that we could start tackling. And I think we've just started tackling cancer over the first few years, and now we're moving to neurological disorders, to cardiovascular diseases. There are so many options that we can work together and opportunities to collaborate to do more.
Yeah, my answer is a little bit different kind of project, but one of the things that I've been working on over the last several years is trying to make it so that our clinical collaborators, whenever they have sort of one of these ideas for an app or some data that they could collect that would be helpful in their clinical workflow, how can we make it as easy as possible to get those data into the hands or into the hands of the care team, the researchers, or the patients, right? And we've spent a lot of time working on these problems with ResearchKit and with ResearchStack. Again, this Common Health app is an example of this. But in the end, there's still challenges because everybody's needs are slightly different. And you go and you try and work with building, you try and build an app for a given clinician, and it's just a little bit different than the one you want to build for the next. And then, never mind the fact when you try and leave this institution and go to another, the challenges between IRB and contracting and vendors and all of those kinds of things. So my real wish is that we could have a scalable opportunity, a scalable way to deliver these technologies to the clinicians trying to do early stage research in a way that's affordable and manageable and easy.
So we recently worked on projects that the goal of projects is to classify the accuracy versus non-accuracy prostate cancer based on the MRI images to prevent the unnecessary biopsy. Presumably the biopsy is very painful, and people goes to the biopsy 2 to 3% statistically shows that they got infection. So and because it's very risky, physicians send them to pathology because this is the most accurate way to detect aggressive cancer through these patients. So we try to detect the aggressive versus non-aggressive patients for prostate cancer based on the MRI images, but these MRI images labeled based on the pathology. So we thought maybe we can train an algorithms that can detect something through these MRI images that the human eye cannot detect it. So my dream project is to extend these methods to other type of the cancer, like lung cancer or breast cancer, which is, I'm pretty sure that for these type of the cancer, biopsy is very painful. And so the main goal, the ultimate goal is just prevent unnecessary biopsy, which can reduce the cost and also pain.
A question, and maybe it applies to the rest of you as well, is that when you do that, what will make you trust it? What do we need to do in our processes as we identify some of these things to then be able to explain and trust that? And how do we have to be careful in the data we're using to drive the models?
So for our projects, we compared the result. First of all, we extracted the test set through from the image source as a blind test. So we know that the algorithm, we just measure the performance of the algorithm for the blind test. Second, we compare these results with the with the score that usually the uroradiologists use it, that they call it PIRAT. And we found that based on the Cohen-Kappa measures, we found that the high correlation between our algorithms result with pathology, how much is higher than the correlation between the agreement between the PIRATs with pathology. So we know that at least the algorithm can do better than the PIRAT score, which is usually used currently for MRI.
Do you guys want to comment? Anything else that you want to?
I think one of the key components is transparency, especially when you're talking about AI. In order for people to trust it, you need to also show the materials and methods and the approach that you're using in order to come to that conclusion. So building that trust, I think it's important on a scientific community that you provide this and set it to stage for people to be able to evaluate. We cannot give our data because there's also the privacy concern that we should factor that in, but making sure that this is out there available for everyone to improve upon or give feedback, I think that's a key element.
And also, the algorithm can be a second opinion, help physicians, so it works to work on that, yeah.
Maybe now we can ask you guys if you have any reactions, any reactions from the audience regarding this discussion, any questions that you have of the panel. Yeah, Cora.
How as we as clinicians interact more with you? How
Yeah,
can we do this?
excellent question. Do you guys?
By one of them.
Yeah, so nearly every project that we work on is, or at least for myself, comes from a clinical need or question, and it often happens in conversations at meetings like this. There are certainly times where someone gets referred, right? Such and such wanted to do a project, and they know that you're the guy that builds X, or they know that Deborah does Y, and she's too busy, and so people end up emailing me. But seriously, I think that understanding enough, right, from the clinical side, understanding enough about what the capabilities of the technology are, that you can at least ask the right questions of the technology experts, I think is extremely helpful. There are often technologists who have this sort of like hammer and nail problem that are going around looking for clinical domains, and we certainly have done some of that as well. So just having enough of these kinds of conversations that you understand the technologies that are available and thinking about ways that they might apply to your domain is probably most successful. We try and have these once a quarter or once a semester meetups, which are okay attended, but I think a lot has actually come from them, and so that sort of thing is super helpful.
So just for our own education, so those meetups happen at Cornell Tech or?
Both.
Okay. I don't think we knew about those, so I think it'd be great to
They
publicize
happened
them.
last year, and I think they were just neglected
Okay.
to be scheduled for this year. But
Yeah.
yeah, or figure out some other way to do them even better. Because I think it would just be fine to just have a session where people who are interested come and talk, and we narrow it a little bit, because I think it's a little hard. This is a pretty dynamic range that's a little broad, but we can cycle between them. So here are a couple people who, this is my type of hammer, and then anyone who has possible clinical interest that matches that tool, we can have a conversation. I think would be an even more useful format than wine and cheese.
I think that's wonderful. We've come up with our first action item from this from this symposium, so that's wonderful. Thank you, Cora, for bringing this up. Any other reactions from the audience regarding technologies for clinical care? Any? Yeah.
Mine is more of a comment and a perspective to how to implement this new technology in scientific research into the clinical world. And I guess this comes from my frustration over the years in trying to implement into the clinical realm what we're doing. We've been successful, but it was, I think the big elephant in the room is the IT department most of the time, that, they follow different rules, different, compliance and privacy, safety, et cetera. And do you have any comment on that? Do you think that this is, having a more research-oriented IT departments in hospitals might be, a way to go.
I will answer that carefully because I spent the last couple of years dealing with these exact groups at probably 20 different institutions. I started a couple of years ago, started a company called Curiosity Health, and the idea of it was basically to deliver apps and services to clinicians looking to conduct early stage research. And ultimately, we've gone in a different direction, and a lot of that was because every time you go to a new institution, a new researcher would like to start a project, you go through the exact same process and go through the exact same vendor onboarding and the exact same security audits and all these sorts of things. And some of the institutions that we were brought into as a vendor actually had groups that were doing this kind of work, and the clinical team didn't even know that that group existed, right? We here have the CTSC at Cornell, which does certain things very well, certain things that they're not really set up to do, but there has to be some sort of model, right, where it's either easier for outside vendors to come in and do this work for you, or the tools in-house exist. Otherwise, it's just not feasible on an R21 budget or less to go and build the kinds of services that we're talking about.
I think one of the challenges that every PI faces is when they're pushing the boundaries and creating something new, they often hit that wall that's called IT that says, pick from the list what you want to do, and they often see their research not on the drop down. And we faced this with Andrea when we were integrating our genomic data to our EMR, where the question was, how many columns do you need to put exome sequencing data? And at that time, it wasn't mature enough, but we did, sure enough, find ways to go around this and provide our clinicians with the full data set. I think it's always going to be a challenge when you're pushing the envelope and getting IT to learn to relax the rigidness, but also maintain that security that we care for. And maintaining the privacy is kind of a difficult thing to juggle and always be patient as they are learning most of the process with us along the way.
Maria, yeah. Okay, we have to wrap up. Well, look, it's been I think it's been a great discussion, very informative. We have at least one action item, which is to not have any wine and cheese, but have meetups instead. Thank you, Deborah.
But we have a working group session.
And we have a working group session, coming up a bit later. Well, we will go into more details about a lot of these aspects and come up with recommendations, with ideas and suggestions for the future. So I want to thank everybody in the panel for all the great discussion, and thank you, Deborah, also for being a great co-chair.
Alicia's not here? Is Alicia here? You have this? Huh? You have it? Huh? That's the same, huh? So, go ahead. Okay, so you've all met me yesterday. I'm Cora Sternberg. I'm the clinical director of Eipm, and I'm co-chairing this session called Translational Research Together with Alicia Alonso. I'd like you to introduce yourself.
On the other corner, it's Alicia. I'm the director.
Use the
I'm the
microphone.
director of the I'm the director for innovation for Eipm. I'm also the director of the epigenomics core, and I'm a molecular biologist. So my innovation is mostly genomics and epigenomics, but I'm willing to cross boundaries whenever necessary. So our panelists. Shall we start from over there?
You want to
I think I
introduce yourself and highlight the programs you're part of?
Yeah, I think I already presented this morning, so I shouldn't explain too
we
much.
all know you. I think
We know.
met,
Yeah,
right?
yeah. No, okay. I think
Talking
there are
about,
people coming later. So I'm Carsten Sucher. I'm
work across campuses. We have met, but we've never really done anything together.
exactly.
Although
I'm,
we like epigenomics.
yeah, I'm from the Qatar campus, and we are working on all kind of omics over there, including, like you mentioned it, we also have methylation. So we have the genomics core run by Joel Malek, so we can run RNA-seq, DNA-seq, methylation 450K arrays, things like that. And then we have a lot of other omics like Metabolon platform, Biocritus Metabolomics platform. We had a Homologic that is discontinued now. And quite recently, we also got an Olink platform. So I think we can provide core services to whatever you might like to use here. And I kept with it. All right?
Great. I'm Ben Coatesgrove, an assistant professor in biomedical engineering up at the Ithaca campus. I work with Olivier's group as well as groups in the Hospital for Special Surgery here in orthopedics and plastic surgery in Weill Cornell to look at skeletal muscle repair and regeneration, and broadly our group's interested in the interface between single-cell genomics and tissue repair in the orthopedic system. And we have students here that have bridged that gap between the two campuses, and I'm happy to talk about some of those efforts, both from the graduate student perspective, but as well from other consortium that have spanned our two campuses.
Thanks. I'm Ankur Singh. I'm an associate professor of mechanical engineering and biomedical engineering at Cornell Ithaca. I'm an immune engineer. I build materials that interact with the immune system to control their behavior, to study them. We have been building several ex vivo models, and that's been some of our key connections with the Cornell medicine side. I work closely with Ari Melnick, Olivier Elemento, Giorgio Ingarami, and have several other projects going on with Monica Guzman and some other key collaborators over here. So, I have a student who has been here more than 1 year with Olivier now, and I think that's been one of the key steps we have taken to bridge the gaps over here, and it's really the results come out to be very, very promising from there. So like Ben, I'm happy to talk about that as well. I'm also a core member of the Englander Institute, and I think that's been very, very useful for us bridging the gaps and working on several aspects of it.
Hello, everyone. I am Laura Martin. I am leading the organoid platform at Eipm. We grow organoids from different kinds of tumors, solid tumors, and we would like to expand our collaborations across campuses. And I agree with you, Ankur, having students coming across different campuses, I think that helps bridging the gap.
Okay. So we're talking about working across campuses. How do we meet and start to collaborate with the other groups? Any of you have any suggestions how we can do this?
I can say that I started my efforts before I even moved to Cornell, and my first grant was submitted to the NIH 15 days before I arrived on Cornell campus. So I found it very collaborative because what you have to do is you have to take the initiative and reach out to the people on the other side and then brainstorm from there. So. And since then, I've never stopped. I continue to reach out to various investigators with some thoughts based on what I've seen, and they're very willing to interact. So if you're not proactive about this, then the collaboration may not happen. But the easiest thing to do is do a little bit of research, have ideas that you want to pursue, and then you reach out to these collaborators. In most cases, it works out. Sometimes it doesn't, but then you can always go back and figure that out.
What kind of resources do we need to do this, to really work among campuses, Qatar, Ithaca, Cornell Tech here?
One of the things that has nucleated several new ideas that I've seen over the years is this cross-campus seed funding effort. So when I came to Cornell in 2013, at that time there was a funding mechanism, about $50,000 seed funding that was removed a couple of years later, but then a newer initiative has come out which is even substantial than that. I think having these kind of initiatives really helps both sides to have equal interest in that and nucleate new ideas. So I think that's one thing. And having these kind of symposium actually leads to cross-collaborative efforts.
How easy is it to actually share a student? across campuses.
Maybe I can talk a little bit about that. So again, Ankar and I are both affiliated with biomedical engineering, and just for those clinicians in the audience, the type of students that we have span from computational biology to people who are interested in building organoids to people who are interested in digital health platforms. So a broad set of students. And the students that we have are housed in Ithaca, but many of those students come down and complete their PhD here in the city in some sort of clinical or engineering lab. Those students typically have come down for the duration of their PhD, but many students actually find that short-term stays are beneficial for their work. And again, Ankar and I both have students that are housed in Olivier's lab with, I think, great success in terms of building the collaborations and nucleating very practical solutions to the trans-campus problems. One of the, I think, on a personal perspective, one of the things that has been beneficial is that the graduate schools between the campuses have worked out better mechanisms through the linkage program and through better understanding of the expectations and the funding of graduate students to make the sort of administrative elements much more streamlined. But there are still practical things about housing, like how does a student come down for a month rather than a whole year, that are challenges that are partially going to be addressed by some of the building at Cornell Tech. I think these sort of things, though, are ones that we as faculty don't often see, so I think students can rise some of those issues to priority and to importance. But I think one thing that we often miss is that the students really lead the efforts. And so things like our program's immersion program, which brings students into the clinic here, really help build an on-the-ground presence between the two campuses. And I think there are additional practical and logistical things that I think broadly could be addressed to help make those things a little more productive and a little more streamlined.
I have a question though. Do you have, I don't have knowledge. Is there any like shared PhD program you can have like two mentors from different campuses?
It's a good idea.
So there's not as much formal, although the Trans-Campus Tri-I program has historically involved students that are housed here to go up to Ithaca and then split their student composition at the graduate level between the two campuses. I think increasingly people like Olivier and other people in the audience perhaps here at Cornell are actually faculty within our, for example, biomedical engineering program. I think there's a few computational biology as well. So in cases where there's formal affiliation, then those faculty can serve as mentors or thesis committee members, certainly.
And these affiliations are relatively straightforward approaches. If you have expertise that fit into a particular department, so I speak on behalf of also mechanical and aerospace engineering, and we have several biomedical faculty who are affiliated with that. We have a mechanical engineering faculty who is also a professor of medicine at Cornell, while Cornell. So depending on that, you could be nominated, you could become a field member of that, and some fields have some requirements, others don't have as much requirement. But that really facilitates you taking some of the students if you're interested or co-advising efforts. Yeah.
Are any of the students going to Qatar from here?
I would say it's the other way around. I mean, yes. Most of our students come here to New York. And I think one model would be, for instance, to mirror things that exist here in New York. In Qatar, we have an example, like the team from Sudan, the kidney transplantation. There are 2 residents from his group who returned to Qatar, and we are now implementing the same IRB over in Qatar to do sample the same size, doing the same things and stuff like that. And a parallel thing is what we now try to do here with the cancer. Like a member from my group tries to get an IRB to collect tumor samples freshly, do metabolomics on them, and together with my group here with Jan Krumzig, not my group, his group, sorry, is to make sure that we maybe do the same thing, to collect tumor samples in both campuses and make things comparable. But I think the other thing is also the important message is like we have a visiting scientist program in Qatar, so we can also host students. I actually did it already in the past with my home university in Germany to say we do a joint project and then students can spend half a month or 2 months in Qatar and then come back to the US. And that would be really something we could support from Qatar also financially to make sure that their finances are covered and things like that.
Do you have patients there in Qatar? And do you have patients in Ithaca? Or do you have access to clinical patients?
Well, the clinical patient access is largely through Weill Cornell, from here. And we have had patient samples shipped right after surgery to us, and And we have sent students down here. If there is something that cannot be shipped for some reason, then students come down here and work, but no patient as much.
Yeah, actually, we don't have to talk about patients. I just learned that there's an animal biobank in Ithaca,
Yes.
and we also collaborate already with the Baker Institute on horses. And I think, especially in terms of animals, there's also a lot of cross interest between the
Well,
companies.
I agree with that actually, because I work with both the veterinary school side of things and the medicine over here. So for some of our lymphoma projects, we have these dog patients that we have worked with. We have generated with the experts over there dog PDXs that we have run studies on. And of course, from Giorgio Ingrami over here is leading the PDX efforts. So the initial PDXs that are generated are then transported to Ithaca, and then we have a full set of PDX studies. So it's very, any human sample comes from this side for us at least, but there is a substantial biobank and other resources at the animal interface. And it goes from small animal models to very large animal models, and it depends on what your need is.
It seems to me as a clinician, humble oncologist, that we actually need each other and can work better together. I mean, really need each other. You have expertise in Ithaca and Qatar and other places that we don't have, and we have patients in clinical trials, or we can invent clinical trials. And it worked. It's easy to work with Laura, who's here, but I think we have to find a way to work across campuses. I think
And some
really
of
important.
and some of that people have figured out. So when I started, the current provost was the dean of vet school, Mike Kotlikov, and at that time, Weill Cornell and Cornell veterinary side teamed up to initiate a PATH program, and that led to a very nice grant from Lymphoma Leukemia Society that integrated both the human and the animal side. So there was a clinical trial kind of efforts related to the canine models that Christy Richards was leading at the time. So yes, I mean, those are successful examples to look forward to and build some of the future work.
No, I was going to say, how are we doing on time? We're not keeping it. I was going to say, because there's a lot of different projects. The question is, where are we going as a community, I guess? And I think it's open to the forum. Like, where should we put our efforts?
Where should we put our efforts?
Yeah.
Should it be on precision medicine?
Should there be a, yeah,
Should it be on precision
concerted
medicine?
effort, or should
What do we
just
think of
everybody go
precision
on his own?
medicine? Does anyone want to take a stab at that one?
'Cause, I don't know. Should we be very varied, or should we focus on something and try to make a mark? I think it is actually what people are asking about precision medicine over and over and over. So go for it.
Well, I think Olivier had maybe mentioned this earlier, but I think seeing the Precision Medicine Institute expand away from cancer into other areas. I obviously have my biases in regenerative medicine and orthopedics, but as we heard earlier, maybe also pushing this into more physiological questions pertaining to metabolism and exercise and rehabilitation sciences could be benefited from a precision or personalized medicine type of an approach. And there's already the infrastructure to facilitate some of those things. Some of that expertise, at least here, is housed over at HSS and other places somewhat more locally, but there's strong connections at the graduate school level and various collaborations between the campuses. Actually, there's a very long-standing set of collaborations between Onkar's department back in Ithaca in mechanical engineering and the orthopedics program here at HSS, or in a neighboring, I guess, site here at HSS. So I think seeing these examples push forward into other areas might be the next step.
I think one thing that can further streamline, I just want to bring this up because I think it's important. So, and I brought this up to Olivia at some point too, that working with the Precision Medicine Institute is great. And there is enormous amount of data that's available that we can, as investigator, mine through that and come up with our own hypothesis projects that we can team up and lead. I think facilitating some of the access to that data is currently, at least to my lab, is not accessible. So we reach out back to Olivia for help. But if it is accessible to select people, we can have a platform like that. NIH has a DGAP program that allows you to get permission from them to download some of the data set, and it has various rules and regulations related to that. We could do that, but allowing us to access that data will help us come up with our own projects that will further nucleate new ideas over here and reduce the burden on the EIPM team as well, I guess.
Laura, you want to say something?
Yes, from my perspective, from the organoid platform, right, we have models of different kinds of tumors, and that's a renewable resource, right, up to a certain extent, but it can be exploited. You can develop new drugs, new combination of drugs. We have the high throughput drug screening platform, and it's almost ready. And we have the organoids, right? So we can focus on every disease, any question, right? Mechanistic studies, new drugs, but then also from our like more precision medicine point of view, we would like to, and we are collaborating with Anchor in this regard, to bring these models closer to the patient, right? They are already similar, and we claim that these are a very good model that recapitulates the disease, but we are still missing components of the microenvironment, specific components of the extracellular matrix, and I think that's where we are going now. That's the direction that we are going.
Do we have any comments from the audience? Yep. Anyone want to say something over there? Here, I'll bring you a microphone.
Yeah, I just have one question. In terms of translating this, I think what we hear is there are torrents of data being generated for everybody. But one of the big issues here is that for the majority of the population, these torrents of data might not really be feasible. And so in terms of how to translate this, how much effort is going into taking these observations on these massive data sets and then going back to very simple clinical tests that could maybe bring that knowledge that's coming out of this to community clinics and places where people aren't going to get organoid technology and NGS. I mean, how do you do that? How do you make this something that's applicable generally?
I think so far what we have experienced is that we have not gone that far out yet, but we have helped investigators over here to come up with better drug parameters that allow them to design their clinical trials. One of the we have a new grant or one coming in with Ari Melnick that has helped aspects that's leading to this particular new drug that was developed and the clinical trial in seeable future over there. And that's for how I see, and that's as a contribution from my side, the technologies that we are building that can have, and so it's quite possible in that I think in coming years it should go to the point that you're referring to.
I think that from Weill Cornell point of view, from the medical center point of view, and we have we're now in Brooklyn and in Queens as well too. We have various centers. I think that if we can begin to work with you on some clinical trials and to understand better how to treat patients, that will then come out into the community. But I don't really see how we can start in the community if we don't first start working here together in this network in a better way. I think a meeting like this is really important, so we get to know each other and understand what everyone is doing, and we can develop relationships. Do you want to say something, anyone? Alicia? No. Anyone else?
I've been in multiple institutions with these smaller planning grants, which I think are great, but I'm wondering if we can try to go for something a little bigger with follow-through. So if we somehow had these smaller things, but with some clustering, so that maybe they could wrap up to generate evidence that goes into some larger scale proposal, it would
work together to write a grant together.
Yeah, and that these targeted proposals, their intention would be to try to get some preliminary results that bring things together, so it's even beyond the scale of 2 collaborators. There's some cluster that's a little bit larger, so we start having taking advantage of all this work.
I think that would be great.
Yeah, and I think there are initiatives already now moving forward with the inter-campus seed proposals that are there. There are two mechanisms. One is multi-PIRO1s. The other one is more like a Center P grant or U grant mechanism. And they provide you up to about, if I'm not wrong, $200K over 2 years. And the idea is to bring all campuses together and initiate some ideas. So there are some things going on that several people over here.
Maybe we should have that as an action item that
Yeah,
we're all very
Yes.
interested in knowing about.
Yes,
more
so
about
new call is out just now. So we have had 2 years of successful program over there, and
You know about this?
but it requires you to have people, if you're working between 2 or 3 campuses, it requires you to have those people over there and there are funds allocated for those. So yes, I will do that.
Okay, so that's the action item from this panel. Thank you very much, all of you. I think we have to go on to the next group meetings. Thank you.
Thank you.